Origin of organized activities of life. How had life come to use DNA? Origin of activities of life. Early Earth's ocean had been carbonated water. In the early Earth, 4 billion years ago, when liquid oceans were formed, the concentration of carbon dioxide (CO2) in the atmosphere was 10,000 times higher than today. When the seawater temperature drops, a large amount of CO2 molecules in the atmosphere dissolve in the seawater. At that time, the CO2 molecule has a linear shape with a carbon atom C surrounded by two large oxygen atoms, so it fits into the through hole of the helical structure formed by the molecules of hydrogen-bonded liquid water. Formation of the helical structure of water molecules in carbonated water. The molecular arrangement of liquid water molecules in tetrahedral units varies in time in units of 10 to the minus 12 seconds, making it difficult to observe. However, the molecular arrangement of the three-dimensional structure of the tetrahedral unit has been investigated in detail for SiO2. In general, the smallest molecular sequence of the smallest energy in a tetrahedral unit is the helical structure of alpha crystal. The negatively charged CO2 fits into the through holes of the molecular arrangement of the helical structure constituted of hydrogen-bonded water molecules, as shown in Fig. 1. Origin of vital activity. It is unlikely that continuous vital activity arose suddenly. It is thought that the membrane formed by gathering hydrophobic molecules in water creates a boundary with the environment and initiates life activities. Hydrophobic hydrocarbon molecules float on the surface of the water and gather to form an oil film. When light molecules generated in water reach the oil slick, vesicles encased in the oil film are produced. We believe that the origin of life activity lies in the thermal movement of the molecules of the vesicles. Respiratory exercise was generated by coordinated rotational motion of tetrahedral units in helical structure. The helical structure of alpha quartz is a structure in which the electrical axis is alternately rotated by an angle phi in the molecular arrangement of the helical structure of beta quartz as shown in Fig. 2. In the transition from a beta quartz to alpha quartz due to this rotation phi, the reduction factor in the z-axis j direction is gamma z equals cosine phi, but in the xy plane gamma xy equals 1 plus square root of 3 times cosine phi, divided by 1 plus square root of 3. In alpha quartz, the rotation angle is about 17 degrees. Evidence that the helical structure of water is formed in contact with a membrane of bubbles in carbonated water. In the thawed state, the thermal motion of molecules is not intense. We can observe how the small bubbles that are close to each other connect, and the move will be shown the next. The linkage of this motion is thought to be caused by the presence of a low energy state that is reduced in contact with the membrane of each bubble. In other words, the linked or synchronized movements of bubbles is the evidence that the helical structure of water is formed on the membrane of bubbles. Coordinated motion associated with rotational motion of tetrahedral units of a helical structure. As shown in Fig. 4, in the organization of water molecules in the helical structure, expansion and contraction movements occur due to the rotational motion of coordinated tetrahedral units. Along with the thermal movement of the helical structure of the liquid water molecules facing the membrane that repeatedly expands and contracts, the molecular organization of the vesicle membrane also undergoes the movement of expansion and contraction. Therefore, if there is a direction in the molecules that pass through the membrane structure of the vesicle, the molecules that are beneficial to the vital activity of the vesicles are taken in, and the molecules that are disadvantageous to the vital activities of the vesicles are expelled from the vesicle. However, as a life activity, it is necessary to maintain a coordinated movement of expansion and contraction. Maintenance of lifespan by the generation of components of the vesicle membrane. Here, we show that the molecular structure of hydrogen bonds can maintain their lifespan through metabolism. When a fine powder of iron is added to carbonated water, air bubbles are generated as shown in Fig. 5. This bubble is formed by hydrogen bonding of bicarbonate ions HCO3 of iron bicarbonate generated by the reaction shown in equation 1 when a fine powder of iron is dissolved in liquid water. This bubble does not break even if you hit the glass container and shake it. Rather, it is easier for the bubbles to break if you stop hitting the container. This is evidence that the production of vesicle membrane components is effective in preserving the life of vesicles. Origin of CO2 and H2O seawater of the Earth. CO2 in the Earth's atmosphere is solid dry ice, and H2O in seawater is thought to have been involved in the birth of the Earth. Currently, the Sun collects 99.86% of the total mass of the solar system, and 0.14% of the material in the entire solar system is made up of planets. Therefore, it is thought that the Earth was born in a cold environment before the Sun's nuclear fusion reaction, and the interstellar medium was formed at the same time as the Sun. The Sun was located in the center of the solar system and rapidly grew into a huge size. The planets grew by collecting interstellar matter that was in the same orbit of the Sun, so they could not grow rapidly and largely. The Sun has grown so much that it can collect hydrogen like Jupiter, and the internal pressure has increased and become hotter, and the internal structure has progressed. H2O and CO2 in the interior of the planet were also degassed to form the primordial atmosphere. On the other hand, when the Sun became stronger and exploded by nuclear fusion, a large amount of material that formed the core of the Sun was released as an interstellar medium or meteorite. The impact of the meteorite heated the surface layer of the Earth, and CO2 and H2O were added to the composition of the Earth's atmosphere by degassing. Reactions of solar wind H+ with Earth's atmosphere. The explanation that the Earth is protected from the solar wind by the geomagnetic field needs to be reconsidered. The magnetic field lines indicate the direction in which magnetism works. The bearer of magnetism is charged particles in motion, and magnetism is a short-range force that decreases with the square of the distance. The charged particles in parallel motion have a magnetic coupling, and the motions of the charged particles are chained. Evidence of the solar wind and the Earth's atmosphere at action is shown in Fig. 7b. In other words, the lunar orbiter, Kaguya, has observed high-energy oxygen O+ only when it crosses the Earth's shadow with the Moon. Formation of cell membrane at the early Earth. As shown in Fig. 8, the cell membrane consists of 3 O groups of glycerol C3 hydrogen oxide and 2 fatty acids ester bonded to form a hydrophobic part. A phosphate group PO is attached to the remaining one, and the polar group X is further bonded to the phosphoric acid to form a hydrophilic head. Long-chain hydrocarbon molecules become liquid, and when they fall to the surface of the water, they float and concentrate on the surface of the water. The cell membrane of living organisms on Earth consists of long hydrocarbons C16H34 or C18H38. This substance cannot be easily synthesized by chemical reactions in water. As shown in Table 1, C16H34 and C18H38 were in a liquid state in the temperature range of the Earth's surface in ancient times and floated on the surface of the water to form hydrophobic membranes. Evolution of cell membrane. Formation of vesicles by fatty acids. When floating long-chain hydrocarbon molecules are irradiated with ultraviolet rays from the Sun, fatty acid molecules are produced with oxygen attached to the ends of the hydrocarbons. A membrane of fatty acid molecules is produced that passes through the molecules in only one direction. Vesicles are formed with the hydrophilic head of the fatty acid molecule on the outside and the hydrophobic tail on the inside. Synthesis of proteins by amino acids with side chains attached to cell membranes. As shown in Fig. 9, the cell membrane consists of 3 O groups of glycerol C3 hydrogen oxide and 2 fatty acids. The hydrophobic part is formed by ester bond. A phosphate group is attached to the remaining one, and the polar group X is further bonded to the phosphoric acid to form a hydrophilic head. On the other hand, the side chain R group of the amino acid molecule has the property of adhering to the cell membrane. The cell membrane that incorporates the molecular sequence of amino acids changes under the influence of amino acids. Structural proteins are formed by combining amino acids and cell membranes. When the chain reaction generated in the cell membrane and the molecular sequence of amino acids are combined, enzyme proteins are formed. Cell membranes whose structure has changed by incorporating the molecular sequence of these homochiral amino acids are involved in protein replication. Control of protein replication by molecules of right-handed L-type and left-handed D-type amino acids. As shown in Fig. 10, proteins are peptide-bonded amino acid molecules of the same chirality. Therefore, 2 single-stranded proteins of different chirality are formed. Watson and Crick DNA helix structure is shown in Fig. 11. 2 helical structures with different chirality in DNA do not mix, and travel opposed directions. In the molecular sequence of 2 proteins paired with L-type and D-type amino acids of the same amino acid, molecules of amino acids of different chirality have mirror image surfaces of object and are the same size, so that the molecules of the paired heterochirality amino acids can be sorted in the amino acid step. Under this condition, amino acid molecules of proteins of the same heterochirality are added to the amino acid sequence. How did life come to have DNA? In creatures on Earth, L amino acids bound to tRNA for protein production are linked on liposomes according to mRNA, which is a polymer of nucleotides with pentasaccharides of D-type chirality. Here, control cannot be established unless the thing to be controlled and the thing to be controlled exist at the same time. Proteins of L-type amino acid polypeptides and mRNAs of D-type sugars, which have different chirality, can be controlled by each other without mixing. DNA is composed of complementary base pairs of L-type nucleotides and D-type nucleotides by hydrogen bonding, as shown on the left side of Fig. 12. The processing of replication, so-called central dogma. Replication of protein takes place in the order known as the central dogma. That is, DNA to RNA, RNA to protein. On the other hand, the organization of molecules that replicate proteins evolved in the order of RNA tissue from protein tissue and DNA from RNA tissue. In DNA, as shown in Fig. 13, pairs of homochiral amino acid molecules with chelations that replicate proteins and molecules with heterochiral chirality amino acids are replaced with stable hydrogen-bonded compensated base pairs. Therefore, when the double helical structure of DNA is separated, base pairs of different original chirality are generated, and one can be matched as a template for the other. At that time, the progression of the reading strand and the lagging strand is reversed and the operation is performed in the step of the amino acid molecule. How to replicate DNA by ushing heterochiral RNA. To replicate DNA, the double helical structure is separated by a helicase into a leading strand and a compensating string connected to a lagging strand, and each is replicated into two strands of DNA. In replication using the leading strand as shown in Fig. 14, L-type nucleotides are added in the opposite direction to the replication fork progression, so the synthesis reaction proceeds discontinuously at amino acid unit breaks and proceeds back from the replication origin. Moreover, amino acid molecules are specified by 3 sets of compensated base pairs. The replication process is carried out in units of 3 sets of compensated base pairs. Summary: more than 4 billion years ago, the Earth's atmosphere was composed of thick carbon dioxide, so a large amount of carbon dioxide was dissolved in the ocean water, and carbon dioxide molecules were contained in the through holes of the helical structure of water, and the helical structure of water was coordinated in thermal motion. There is no doubt that the origin of activity of living organisms had been derived from the thermal movement of liquid water molecules in 3 dimensions by hydrogen bonds. Regarding the formation of long chain of hydrocarbon molecules, which are the main components of the cell membranes of living organisms on Earth, hydrocarbons were produced by CO2 in the primordial atmosphere and H+ of hydrogen ions in the solar wind, and explained that the conventional theory that ignores the influence of the solar wind. Furthermore, it is pointed out that the role played by the chirality of the molecules involved in the structure of DNA from the structure of DNA. That is, heterochiral molecules are the same size even if they have different chirality, and they have mirror image inversion symmetry faces, so heterochiral molecules can be selected by matching them to the molecules in the template. Therefore, it cannot be easily converted into molecule of different chirality, but it can be generated by selecting and adding heterochiral molecules sequentially. In this way, it had taken many processes to establish a mechanism for living organisms to replicate proteins using DNA, and a full-fledged biological mechanism was established. Thank you for your attention.